All right, what you're looking at in front of you is a picture of a eukaryotic chromosome. Again, prokaryotes and eukaryotes we discussed a couple times so far this year. The major difference between prokaryotic and eukaryotic cells are prokaryotic cells do not have a nucleus, and eukaryotic cells do have, each eukaryotic cell has its own nucleus. And what you're looking at in front of you is a picture of a eukaryotic chromosome. Looks to you as a giant X, which is all that different from what it actually looks like. If you put it under the microscope, it'd be less colorful, but it would look pretty much the same. And basically what you're looking at is a giant X, which the left side of the X is one of the, quote, sister chromatids, and we're going to get to that later. And on the right, on the right is another sister chromatid. Again, two different chromatids combined at one point where the X meets itself. So think of them as two different chromatids, and these two actually become separated upon mitosis, which, as we'll learn shortly, is the process by which cells divide. So what you're looking at again here is a picture of a chromosome, one chromosome, two sister chromatids, and this is basically a picture of a eukaryotic chromosome. Okay. Right, and so if you just follow along on the slides right now, we're going to be going through a couple of slides. You can notice here that DNA is packaged into one long molecule called a chromosome, and basically, what you're looking at, if you see on the lower left section of the X of the chromosome, this over here, right there, that is, it looks like it's a molecule of DNA, and that's actually not that far off because basically DNA is packaged much tighter than it looks in this picture. But DNA is packaged into the chromosome. And if you look on the picture that's fading in right now on the right, you could see a bacterial chromosome is being formed. It's much more simple. It's one ring of DNA. It's much less complicated. It's much less long. That's the bacterial chromosome. Bacteria, again, are prokaryotes. You're looking at the entire prokaryotic cell in this picture, as contrasted with the much larger, much more complicated DNA of a eukaryotic cell. Okay, and in many eukaryotic organisms, there are more than one chromosomes found. In humans, there are 46 chromosomes. This would be one of the 46 chromosomes found in human cells. Okay, we're going to be moving onwards to the next slide. And so the question is, all the way to a chromosome, because DNA is a molecule that you may be familiar with its structure. It kind of looks like this over here. You may be familiar with this double helix structure over here. And the question is, how does it get from looking like this DNA structure here to getting more like this chromosome, which you may be more familiar with on the left side of the screen. The basic approach is, for some terminology, chromatin is basically the terminology we give for DNA and histones. If you look, this is the DNA. This is the DNA right here. It's packaged around some proteins called histones, and it's wrapped into something called a nucleosome. And from there, the nucleosome is a much larger, significantly thicker portion of DNA. It's DNA wrapped around itself. And when DNA coils around histone proteins, that is what we call a nucleosome. If you look as the picture fades to the next section, the nucleosome will coil upon itself over and over again and form supercoils, and that eventually will form a structure similar to the X, which you are familiar with from all the science fiction. Out over here, the third slide, just a little bit of chromosome terminology. So that's the X you just saw before, fading into something else, right? You can see the connection point between the two sister chromatids is called the centromere. And again, as we saw before, chromatin is a combination of DNA and proteins, and that's basically the histones forming around the nucleosomes. Each chromatid is one of the sister chromosomes, and the centromere is the connection point between them. So again, the centromere, the connection point between them. The chromatid is the terminology for one of the sister chromosomes, and over here on the A slot on the left side, you see one chromatid, and on the right